A number of guests duly introduced by fellows beg leave to attend your meeting. I welcome you in your name. Thank you. Thank you all for braving the difficulties of transport, and welcome particularly to the large number of people I gather here online this evening. Minutes.
The minutes of the ordinary meeting of the Society of Antiquaries of London held on Thursday, 3rd of November 2022. Professor Martin Miller, President, in the chair. The minutes of the ordinary meeting of Thursday, 27th October were read and signed. This being a meeting for the election of new fellows, the President announced the following were elected: Marco Imoni, Lee Alston, Catherine Barnett, Cornelius Barton, Christian Koymans, Ross Cook, Roger Curtis, Susan Ghosh, Colin Guy Gilbert-Hunt, James John Morley, Caroline Pudney, Michael Sotherell, Yevgenia Yanish. The certificates of candidates for election were laid before the Society in order to be suspended in the usual manner. The following communication was then laid before the Society wall cap, Hadrian's Wall at the intersection of conservation, research and engagement by Robert Collins FSA. Thanks for return for this communication. The President announced that the next meeting will be on Thursday, 10th November 2022, the adjourned meeting.
Is it your pleasure to sign exactly? Following the results of today's ballot, I declare the following are duly elected fellows of the Society of Antiquaries of London: Thomas Booth, Linda Brady, Matthew Brudnell, Christy Constantopoulos, Matthew Davis, Thomas Del Mar, Alistair Douglas, Christopher Edwards, Henry, Devin Fitzgerald, and Anna Garner. Certificates.
Suzanne Paul, MA, Keeper of Rare Books and Early Manuscripts at Cambridge University Library, and fellow librarian and fellow archivist at St Edmund's College. Liliana Radiovojevic, BA, MA, MSc, PhD, a lecturer in archaeomaterials at the Institute of Archaeology, UCL, and an authority on early metallurgy. Laura Ratcliffe-Warren, BSc, an archaeological conservator with expertise in hazardous materials management and mineralized organic remains. Mark Reagan, BA, a lecturer at Birmingham City University and heritage building consultant with expertise in the ringing and restoration of bells. Steen van Rossum, MA, PhD, curator of Western Religion, University of Amsterdam, and a book historian with expertise in 17th century print culture. Ethan Wottrell, BA, MA, PhD, an associate professor at the Department of Anthropology at Michigan State University, with expertise in the application of digital and computational methods in heritage and archaeology. And Robin Whitaker, MA, an archivist with research interests in diocesan court papers, medieval seals, and the historiography of Worcestershire's antiquaries.
Is it your pleasure that these certificates be suspended in the usual manner? Thank you. We now come to the main business of this evening's meeting, which is to hear our lecture by Professor Ian Armit. Ian studied for his undergraduate and PhD at the Department of Archaeology at the University of Edinburgh. After graduating, he helped establish the Centre for Field Archaeology, now the CFA Archaeology Limited at the university. He left to join Historic Scotland as an inspector of ancient monuments, returning to academia to post as a senior lecturer at Queen's University Belfast in 1999, before moving to Bradford University as Professor of Archaeology in 2006, and thence to University of Leicester Professor in 2018, took up his present post, Chair of Archaeology at the University of York in 2019. Ian's research centres on cultural archaeology of European later Bronze and Iron Ages, with particular focus on the role of conflict and violence in non-state societies, and the demographic and genetic prehistory of European populations. And this is the focus of his talk this evening. He's directed field work and projects in Scotland, France, and Sicily, and has worked extensively in Southeast Europe. He currently is PI on ERC-funded Commios project, again, I think is related to the work this evening. So it's a great pleasure to welcome Ian to talk to us this evening on migration, mobility, and demography in later prehistoric Britain. Welcome.
Thank you very much, and thanks for inviting me this evening, and well done to those of you who've made it here in person, despite the issues that we've all faced today. Just while the screen's descending, what I intend to do this evening, as Martin mentioned, is talk primarily about my current Commios project, which is looking at migration, mobility, demography in later prehistoric Britain, with a focus on some of the recent DNA evidence. Okay. Now, before I get into talking about the Commios project itself, I want to give a little bit of background as to how that project evolved, and give a little sense of why we're currently approaching the work the way that we are. Okay. So to do that, I want to step back a little to a previous project that I was working on with David Reich's team at Harvard Medical School, working on the Beaker complex in the Chalcolithic, the very earliest part of the Bronze Age. And as most of you will be well aware, I'm sure, the Beaker complex is a major marker point in British prehistory in the mid-3rd millennium BC, when we see the introduction of a new cultural package, if you like, across large swathes of Europe, dominated by single inhumation burials, archery equipment, the first gold and copper metallurgy, and of course the beakers themselves. And there's been a debate over the degree to which the Beaker complex represents the migration of people versus the spread of ideas, and that debate really has been going on, for 150 years in archaeology. And this was one of the first big questions in European prehistory that we were able to address through the large-scale use of ancient DNA. Genome-wide ancient DNA analysis, so this is looking across the whole human genome, has advanced hugely in the last, even the last 5 or 6 years, but certainly since 2012, there have been huge advances in analytical methods, in statistical processing, and in the actual capture of DNA. And one of the major turning points really was the realization that the petrous bone, so in your ear effectively, is a fantastic reservoir of ancient DNA, and ossicles within the ear even better when we can get them. So with a combination of these new methods of capture, new methods of processing, the recognition of where to actually source the samples from in the human body, vast advances really were made in terms of the scale and scope of potential ancient DNA analysis that produced a whole swathe of studies and papers from around about 2015, 2016 onwards. I am by no means a DNA specialist, so I'm not going to talk about the detail of any of that really, but my collaborator at Harvard, David Reich, has produced this book, which I would recommend for anyone who wants to follow up on where DNA is at the moment. And basically what we did with the Beaker project was produce a genome-wide study on, at that stage, 400 individuals, and that's 400 individuals from across Europe, so it's a relatively thin spread really across the continent. And it produced really extraordinary results. On the one hand, in mainland Europe, it became clear that the Beaker phenomenon, the Beaker complex, was not associated with any sort of consistent genetic signature. Instead, different communities with very different ancestries adopted Beaker culture. So essentially, it was the movement of ideas. But in Britain, we had a completely different situation. In Britain, we had an extraordinary genetic turnover within the population from the end of the Neolithic into the Chalcolithic, into the Beaker period, that cumulatively, by 2000 BC, replaced about 93% of the genome. So in other words, if you are someone who is living in Britain in 2000 BC, roughly 93% of your genetic ancestors a few hundred years earlier were living in continental Europe. So this was an extraordinary result, confirming that for Britain at least, migration was the key to the emergence of Beaker culture, but on a scale which even proponents of the migration hypothesis hadn't really considered before. And this quote here from a recent book sort of shows part of the reaction to that. However, along with a number of other, this paper came out in 2018, and along with a number of other papers coming out around about that time, the reaction both from the popular press and from archaeologists was one of, let's say, skepticism or wild enthusiasm or something kind of in between. You can imagine the sort of reactions we got in the popular press. The first Britons were wiped out by boozy Beaker people who invaded the UK. Their blood still runs through us today. We've got that kind of thing that we have to deal with, that kind of galloping nonsense really that we see in the popular press. But also from the archaeological community itself, a degree of unease about the way that some of these early DNA papers seem to be kind of galloping away with some of these questions. This idea that we might be seeing this kind of, seeing archaeology becoming kind of subservient to science, seeing the sort of nuance, the nuances that we carefully built up over many generations of archaeological study being kind of wiped away by these great sweeping cross-continental studies. And I think what's happening now really is that we're moving on to the next generation of ancient DNA studies where we're building in a much greater archaeological component right from the very start and starting to look at some more detailed questions. And that really brings us to the Commios project. So this is a project, an ERC project that we began in 2019 to try and harness some of these breakthroughs in ancient DNA analysis and also in isotope analysis, which has been developing very quickly over the same period, and to try and fully integrate them with, or underpin them with traditional cultural archaeological analyses, cultural archaeological questions, drawing on approaches from humanities-centered academics. And that's really what we set out to do with the Commios grant in 2019. We called it Commios. I'm not going to go through all this, but we called it Commios because we're looking at the essentially from around about the Middle Bronze Age through to the end of the Iron Age, and we're looking at links between Britain and the continent and population structure within Britain itself. And Commios is an individual who many of you will recognize from Caesar's Gallic War. He's an individual who's kind of emblematic of those cross-channel links. He's an individual who moves between Gaul and southern Britain, who has great connections in both regions and appears to establish the Southern English dynasty of the Atrebates in sometime after 50 BC. So he's kind of an emblem for our project and a nice acronym as well. You've got to have a nice acronym. Now, the results I'm going to be talking about tonight are not just my work, but they derive from the whole of the Commios team, and these are the main people who are involved in it. I've mentioned David already, Jane Evans, who's running the isotopic analyses, Derek Hamilton, who's running our AMS dating program, and we have 5 postdoctoral researchers working on the project as well, on aspects ranging from the funerary archaeology side of the project through ancient DNA, isotopes, and osteoarchaeology. Again, I'm not going to read all this out, but just to give you an idea of what we're setting out to try to do here. Firstly, we're trying to establish the population structure really of Iron Age, but also of Middle and Late Bronze Age Britain, and examine this in the context of what we would see as traditional cultural boundaries related to settlement patterns, funerary differences, all of those kinds of traditional, I guess, archaeological questions. As well as the DNA, we want to do isotopic studies because we want to look at the degrees of mobility and connectivity within Iron Age communities, as well as their population genetics, as well as their ancestry. And we also want to look much more deeply at individual Iron Age communities, using the potential of the DNA, for example, to identify kinship, familial relationships within Iron Age funerary contexts, and see what that can start to tell us perhaps about, for example, matrilocal, patrilocal marriage patterns, that kind of thing, bringing, I guess, in a way, more anthropological questions into focus. What we started out trying to do, this is Commios in numbers, really. We're now in a position where we can upscale significantly from that Beaker project. The Beaker project looked at 400 individuals roughly from across Europe. Here we're looking at around about 1,250 roughly samples from within Britain itself for this period from around, let's say, 1500 BC to the end of the first millennium BC. Within that, we're targeting a few hundred individuals for more detailed isotopic analysis and trying to build up isotope and osteological biographies of those individuals. And we're able to fill in the dating for around 400 individuals who haven't been previously dated. A lot of the samples we're using actually are from relatively recent excavation and already have direct radiocarbon dates. So we've got an extremely robust dating program. Now, the problem is, this is great, but as you will have noticed, the project started in 2019. And just as we'd recruited all our postdocs, everything shut down. That means that I'm not able to give you anything like the full results of the project. Mostly what I'm going to focus on is actually the DNA, because we were fortunate that we had actually collected a significant number of DNA samples before COVID hit. But what we hadn't been able to do was to follow up to the same degree on the isotopic work, or the osteological work. So that is happening now, but doesn't really feed into the results that I'm able to give you at present. There's sometimes a question as to why we're doing DNA and isotope analysis. Are these not kind of telling us the same thing? But it's important to realize that the two techniques are actually extremely complementary. On the one hand, the DNA analysis is looking at mobility connections within populations at the population scale. So any individual that we sample effectively is not just telling us about that individual, it's telling us about their parents, their grandparents, their great-grandparents, and so on and so forth. So each individual effectively becomes a population in miniature from the perspective of ancient DNA. So we can start to build up complex pictures of biological relatedness between populations and between individuals. And that's where all the statistical processing comes into play. However, the isotope analysis is much more informative on the individual scale, and obviously can give us insights into questions of mobility and diet of specific individuals. It can investigate the life histories of specific individuals. So, for example, if you're you may be a first-generation migrant, let's say, into Britain from somewhere in the continent, and the isotopes and the DNA will both tell you perhaps that that individual has come from somewhere else. As soon as you're a second-generation individual, the isotopes may say that this person is completely local, but the DNA will tell you that actually this person is deriving from a population that's based elsewhere. So the two techniques are really important to tie together if we want to build up more detailed pictures really of what's going on. And it was interesting actually with the Beaker study that previous work with on large-scale isotopic analysis of Beaker populations had suggested that actually there was very little migration from the continent. And that is, of course, because if you don't pick up a first-generation migrant, then the isotopes are not going to tell you anything about movement into Britain. And the chances of picking up a first-generation migrant are, it's tricky to do. Okay. So before I get into the sort of initial results that we had from the Commios study, I need to backtrack a little and explain a little bit about how some of the DNA works. And basically, I suppose the key thing to understand is that for populations in later prehistoric Europe, there are essentially three main components of genetic ancestry, three main kind of blocks of genetic ancestry. The first one is what's usually referred to as Western hunter-gatherer ancestry, which is relatively minimal in most prehistoric populations after the Mesolithic across Europe, but it's still there to varying degrees. The first of the really important ones for us is the EEF, early European farmer ancestry. And this is ancestry that really moves from the Near East across Europe and into Britain at the start of the Neolithic. And around 99% of the ancestry of of British Neolithic populations is what we would call this EEF ancestry. It's coming from the continent. Then there's what's referred to as steppe ancestry, which is derived from populations like the Yamnaya or their neighbors on the Eurasian steppe, which we can see from the ancient DNA moving across Europe in the 3rd millennium BC. And it's that steppe ancestry that comes into Britain in a huge way in the Beaker period. And basically what that means is that all European populations in that sort of Bronze Age, Iron Age period can be characterized by varying proportions of these three ancestry types. It's obviously a lot more complicated than that, but that's the easy way, I think, to try and understand it. And by 2000 BC, so the end of the Beaker period, the genetic signature in Britain for British populations is pretty homogeneous. Almost anyone you sample from around 2000 BC in Britain has essentially the same genetic mixture of these ancestry types. Now, the question that we were initially trying to investigate with the DNA part of the project was this. If you look at England and Wales today, you have a higher early European farmer ancestry than you find in Scotland today. And it's also higher than in the early Bronze Age ancestors of the populations of England and Wales today. So at some point, more early European farmer ancestry has come in to England and Wales, but hasn't penetrated into what is now Scotland. So the question really was why, when, and how did that genetic change occur? We suspected it probably occurred sometime during our Bronze Iron Age period. So with the initial work that we did in Commios, we were able to sample 740 new individuals ranging from the Neolithic to the end of the Iron Age, partly from across Europe, but with a very substantial collection within Britain itself. And at the bottom right of the slide, you can see a sort of simplified time chart. The yellow dots on there are samples that were already published, that already existed. The black dots are the ones that we've collected through that initial Commios DNA work. And you can see that we were very much filling in the period from the Middle Bronze Age through to the end of the Iron Age, which was very sparsely sampled prior to that. So really, and you can also, I think, see perhaps from this from this bottom right image that you know, the numbers are really building up now. The numbers of genome-wide DNA samples are really becoming quite substantial. Okay, so what did this show? Basically, what it showed, and you can see on the graphic here, is that we can see an increase in that early European farmer component of the ancestry in England and Wales over the period from the Middle Bronze Age into the Iron Age, but we don't see it in Scotland. In Scotland, it seems to be pretty flat. And this would appear to imply quite substantial population movement into southern Britain during the Middle to Late Bronze Age. And although it doesn't perhaps look like a huge amount on the graphic here, in terms of the entire genome, it cumulatively represents something like a 50% turnover of the genome in that region between around 1500 and 800 BC. So it's quite substantial. It's not Beaker level substantial, but it's nonetheless quite substantial. Interestingly, it's a little less marked when you look at the Y chromosome, which is inherited from father to son. It's a little less marked there than it is across the genome as a whole. What that might mean is that there may be some sort of slight bias in numbers of females who are moving into Britain in this period. It's a relatively slight, but it's interesting that it's there. It's certainly not a movement of male warriors or invaders coming into southern Britain in this period. That's certainly not what the genetics are telling us. Now, we had to exclude a couple of other possibilities. One of the things that we initially thought, or I initially thought when I saw this data emerging, was that, well, is this perhaps a sort of resurgence of the Neolithic population who had been there prior to the Beaker period? Are these people kind of emerging from the forests, as it were? Had they escaped sampling previously, and are they making their presence felt again genetically later on? But that's not possible. From the genetics. The Iron Age populations of England and Wales can't be explained by statistically mixing groups who lived in the Neolithic and the groups who lived in the early Bronze Age. But they can be explained as a mixture of groups who lived in the early Bronze Age and groups who lived in mainland Europe in the early Bronze Age, and particularly around what is now France. So what it begins to look like is that we're seeing some expansion of population outwards from the region, France, Northern Alpine region, possibly in this middle to late Bronze Age period. It will become obvious. In fact, I may just go back to the map. Yeah. One thing that you'll see that is quite obvious is that there's one horrible big gap on this map, really, that's very relevant to everything that we're seeing, and that is essentially modern France. And that creates a problem because what we can't say for sure is that this is a one-way movement of people from France into southern Britain. It's entirely possible that it's some kind of reciprocal movement, some kind of two-way movement, a homogenization of population in those areas, but we don't yet have the samples from France that we need to really evaluate that argument, although that is something we're very much working on at the moment. Come back to that. Okay, this is another way really of looking at the same thing. What you're seeing here is essentially an indication of the early European fraction of ancestry from the beginning of the Neolithic right through to the end of the Iron Age. And the dotted line is basically giving the kind of mean value of the component of early European ancestry in these populations. And you can see that in the Neolithic, it trots along very, very high. There are some very interesting outliers, but I'm not going to talk about them here today. And then it falls off a cliff, essentially, around about 2400 BC. That is the beginning of the Beaker period, when the genetics completely and very radically change. But then you can see its sort of steady increase through the Middle and Late Bronze Age into the Iron Age. One point to make from this really is that you can see again that the differences we're looking at in the Bronze Age and Iron Age compared to the difference in the Beaker period is very, very subtle. And again, that's where these advances in statistical processing through the Harvard team have really come into play. So let's home in on that period of the Middle Bronze Age to the end of the Iron Age. And you're seeing essentially the same thing again here, that the blue dots are individuals, and you're seeing the essentially the mean of early European farmer ancestry, and you can see that it's rising very sharply there in the late Bronze Age period. But what I want to draw attention to on this image are some of the very interesting outliers that we see, outlying individuals, and that's these ones up here. Because these are individuals who have very high levels of early European farmer ancestry, much higher than we would expect from a British population of this period, and they have it, early. They have it early on. So the possibility from the genetics would seem to be that these individuals may be first-generation migrants. So we were keen to look at these individuals isotopically to see if we could identify whether they had, through their individual life history, moved from a different region into Britain. Now, both of these sites, Margaret's Pit and Cliff's End, are both in Kent, which kind of makes sense if we're thinking about movements from the continent. Both of them excavated by Wessex Archaeology, who've been collaborating with us on the Commios project. And this is just another way of looking at their DNA here, this PCA chart. And what it's showing you is these two individuals from Margaret's Pit. So basically on here, the red dots essentially represent Bronze Age individuals from England. The purple dots, generic ones from Kent. The green dots, ones from Cliff's End Farm that we're going to look at in a minute, and you can see that they're slightly off the general pattern. But these two individuals from Margaret's Pit are very much off the scale here and much more related to individuals of the same period from modern day France and Germany. Now, one of these individuals, unfortunately, had no teeth, so we couldn't do much isotopically, but the other one did, so we were able to look at strontium and oxygen from this individual to try and look at their the region where, give us information on the region where they grew up while their teeth were developing in childhood. And unfortunately, perhaps, this individual marked by the red triangle on here is absolutely smack bang where you would expect her to be. It's an elderly female, exactly where you would expect her to be in the sort of chalk regions of Kent. There's nothing, I mean, this doesn't prove that this woman was local to Kent, but there's nothing to suggest that she wasn't. Now, of course, what does that mean in terms of the DNA? Well, if this woman was the daughter of first-generation migrants, then of course this is what we would see. So it's not really telling us anything there, but it's not getting us any further in terms of thinking about where these populations might have come from. When we go to Cliffsend Farm, the picture is very different. We haven't done strontium and oxygen from the Cliffsend Farm assemblage, but Andrew Millard did this work as part of the publication of that incredible site by Wessex Archaeology back in 2014. And this is a bit of a busy diagram, but essentially what you're seeing here is that box is showing you where the strontium and oxygen ought to sit if these individuals were local to the site of Cliffsend Farm. And you can see that very, very many of them are not. And one of those individuals who's not is one of our clear genetic outliers from the DNA study. So this individual, along with some others from Cliffsend Farm, almost certainly doesn't come from that region. So who was this individual? Well, Cliffsend Farm is an extraordinary late Bronze Age funerary site. The whole range of unusual funerary deposits. The most impressive of which is this late Bronze Age burial pit, which had a series of human remains at various points within its stratigraphy. But at the base of the pit had four individuals, 3 females and 1 male, laid out in this sort of curious, almost funerary tableau. We have DNA results for all of those, and there are no clear biological relationships between them. We can pick up biological relationships clearly to the 3rd degree, so they may be related, 4th degree or more, but they're not close relatives in that sense. This individual is the one who is the clear genetic outlier, and also the one with that isotopic signal that's far from local. And this is a very interesting individual. It's an adult male, dates, well, 912 to 808 BC, and clearly not a primary funerary deposit. In fact, what you've got is the kind of bundled up remains of partly articulated body parts. So you've got the skull, parts of the axial skeleton, parts of the upper limb, and associated with an apparently unique bone and copper object, perhaps a pendant of some kind. So a very unusual deposit, and someone who's got a very unusual isotopic and a very unusual DNA signature. When the site was initially published, Andrew Millard suggested that the oxygen or isotope value, which indicates an area in the blue range on the map here, Andrew Millard suggested that possibly we might be looking at someone who had come from a childhood spent in Scandinavia. The DNA would not suggest that. The enhanced early European farmer ancestry suggests very much northern alpine location when you combine it with the isotopic results. So in this case, we probably have got someone who's a first-generation migrant. But of course, we have a problem because of the nature of this burial. How did this individual actually arrive in Kent? How did he arrive at Cliffsend Farm? This is a body that's clearly been processed, curated, perhaps for a significant period. It's entirely possible that he wasn't even alive when he came to Britain, when he came to Kent. We can't be sure. But clearly this is an individual who was part of that movement of people that we see coming into Britain in the Middle to Late Bronze Age. Stepping back a bit and looking at the archaeology, detecting population movement into southern Britain in this period isn't in itself a great surprise or a great shock. This is a period of general kind of homogenization, perhaps, of material culture in a whole range of ways across broad regions of Europe. So we see, parallel developments in metalwork, in pottery, in settlement forms to some degree, in land use to some degree as well. And we could show any number of maps. This is just a map of bronze pulse stave axes that show these networks, that basically fossilize these networks of contact between Britain and Europe. And French archaeologists working on the Bronze Age have done a great deal of work on these sort of shared cultural units that emerge in this period. The Channel Zone, or Manche-sur-Mer, Manche-Mer du Nord, rather, cultural zone. And one of the interesting things that we see is pottery, parallel developments in pottery, suggesting perhaps contacts at a population level, not just some kind of elite contact that might be represented by developments and movements of bronze artifact types. So that parallel development suggests contacts at quite a deep level, and it suggests contacts that are quite sustained over significant periods of time. We even in this period have started to see the appearance of a number of roundhouses at various sites in northern France, which of course are traditionally associated very much with the distinctive architectural styles of Britain in the Bronze Age and Iron Age. So what we're seeing in the DNA is essentially a population movement that's underlying these kinds of spreads of material culture and these contacts and networks that we see developing in this period. Just another couple of maps really to show the kind of reach of these shared artifact styles, these shared cultural forms that we see in the Late Bronze Age. So there are lots of opportunities within these kinds of networks for people to move around. We can think, for example, about marriage networks. We can think about movements of people in terms of conflict, mercenary activity, perhaps. We seem to be seeing evidence for population growth in this period, which might be pushing people into different regions. There's all kinds of ways in which people might be moving around, and that must be underlying what we see in the genetic evidence. Now, most of our samples numerically were from Britain in this study, but we have got samples, both ones that we've processed and that have been previously published, from a whole range of different regions. And when you stand back and look at those as a group, some interesting trends emerge. So what this graphic is showing you, for example, is that, again, that proportion of early European farmer ancestry, but this time as it changes across different regions of Europe. And when you stand back and look at the overall picture, what we seem to be seeing is a level of genetic convergence, essentially. So, in some areas like England and Wales, we're seeing a significant increase in early European farmer ancestry, whereas if we look at Iberia, Spain and Portugal, for example, we're seeing a significant decrease. What this suggests, I think, is that we're seeing a homogenization of population. So it comes back to the point I made earlier that I don't think we can see this as a population movement, a migration from Europe into southern Britain. I think we have to see this as part of a much broader European picture where people are simply moving around in much greater numbers, and that's having a homogenizing effect on the genetic structure of European populations, certainly North and West European populations in this period. This focus on the Middle to Late Bronze Age is something we hadn't really anticipated right at the start of the project. We were really very much focused on the Iron Age, and we are now doing a lot more work on the population structure within the Iron Age itself. But unfortunately, from my perspective, at least as an Iron Age specialist, the Iron Age has turned out to be pretty boring from a kind of genetic point of view. It's when this movement of population appears to stop. From around, really from the end of the Bronze Age itself, so let's say from around about 800 BC, we seem to see a real stabilization of the early European farmer component around about 800 BC, and no further significant gene flow on a population level during the Iron Age itself. That's not to say we don't have lots of interesting individuals, and you can see some of them dotted around on this graphic here. But in terms of affecting the genetics of the population as a whole, we don't see that change to anything like the same degree during the Iron Age. So what we're not seeing, for example, here is any kind of Celtic migration associated with rattan material culture. Nothing of that kind is making itself felt at a population level. So we see stability with occasional outliers, essentially perhaps individual and small group mobility during the Iron Age, something that we really want to focus in on more. There were other kind of really interesting surprises that came out of the DNA work, simply from the accumulation of large numbers of samples. So things that we weren't really looking for, but the DNA can tell us about different aspects of population genetics. And one of the things that that geneticists are always very interested in is lactase persistence. So the ability to digest milk into adulthood, which is extremely variable across different populations. And one of the things that came out of the data that we accumulated was that lactase persistence seems to increase much earlier and much more rapidly in Britain than it does in other areas of continental Europe. So the red line on this graph, for example, is showing you the lactase persistence frequency in Britain. And you can see it sort of takes off around about 1500 BC, or perhaps even slightly earlier, and carries on really to the end of the Iron Age when it stabilizes. Whereas in Central Europe, it doesn't really take off at all until halfway through the Iron Age, about 500 BC, and it rises to a lower level than it does in Britain. So something very different is happening here. And this is something that appears to be independent of the movement of people from one region to another. This is something that's happening within Britain itself. So like a lot of things with the ancient DNA results, it's kind of throwing up new questions for us, things that we hadn't really thought about before. What is driving this increase in lactase persistence in Britain over this period? Is it some kind of enhanced reliance on dairy products that's happening in the British Bronze Age that we're not seeing in continental Europe? There's also the possibility that it may relate to periods of famine and shortage, where the ability to process a wider range of foods may give an evolutionary boost to particular individuals or small groups within the population. We really don't know yet. It's something that we've been presented with as a question, which we have to then take forward. What's really the focus of our work at the moment? The work I've talked about so far, in terms of the migration into Britain, we've published. I'll briefly mention the paper at the end. But what we're really starting to work on now is the structure and variation within individual communities. Because DNA work at the smaller scale, at the community scale, has extraordinary things to tell us. So, for example, we're looking at double burials in several parts of Britain. This is just one example where osteologically what had been identified as a mother and daughter has turned out genetically to be a father and son, perhaps showing some of the sort of slight unconscious biases in some of the osteological work. But we're finding that these kind of biological relationships are extremely common in double burials. A site that we've looked at in Slovenia, which we're publishing shortly in Antiquity, a site called Dolganiva, we've been able to show that one of these barrows, barrow 1 of the 3 on the map here, which contained 7 burials, is a family group, a father and several of his children and other family members, which is starting to give us some of the ammunition we need to start thinking about how these societies operated in terms of, for example, in this case, patriarchality. I'm not going to go into any depth on this. I just want to sort of highlight some of the ways in which the DNA is beginning to direct us. And just as a teaser almost, we've started to do some large-scale work at the cemetery of Wet Wang Slack in East Yorkshire, which is the largest Iron Age cemetery in Britain, burials of about 450 individuals. And we did an initial pilot study of 90 of those individuals. So we're doing hopefully the entire cemetery over the next few months. But that's showing us a whole web of biological relationships between individuals within the cemetery. This is just one of the sort of family trees that we can abstract from the Wet Wang Slack Cemetery. Sorry, it's a bit complex, but basically anything with a number inside it is an individual that we've actually got from the site. The question marks are individuals that exist, but we don't have their remains yet. The circles are females, squares are males. And this is really exciting stuff because it's showing us we've got 5 generations at least within this family tree of individuals present within the cemetery. And the descent of these individuals is passing through the female line. So we have, we have mother, daughter, granddaughter, and so on, all present. And that's extremely unusual for the few cemeteries that have been looked at so far through DNA in Europe for the Bronze Age and Iron Age, where normally we're seeing indications of patrilineal descent. Here, something very different seems to be happening. There seems to be some kind of real female focus to what we see. But it's preliminary data, and this is something that we're going to do much more work on. And Inigo Ilaldi at Harvard is the person who's really driving this part of our research forward. Okay, it's sort of a whistle-stop tour, and the DNA is pretty complex. So what I would suggest is anyone who wants to read any more in depth on this work, to look at this paper that we published earlier this year that was led by Nick Patterson, part of the Harvard team, who is the brains behind most of the statistical analyses on this DNA work. Well, what can we say in terms of our sort of interim conclusions? Well, what we can definitely say is we've got major population movement in southern Britain in the Middle to Late Bronze Age. And it's not an event, it's a process. It's happening over a period of centuries. The end of that population movement seems to be essentially coterminous with the end of the Bronze Age. So it's behaving itself in terms of how we understand the material culture of the period. The Middle to Late Bronze Age is a period of intense connectivity between Britain and the continent. We're seeing that reflected in the DNA. The Iron Age is not, especially the early Iron Age. And we seem to be seeing that reflected in the DNA as well, with essentially a cessation of population flow across the channel. We don't know yet if this population flow is unidirectional, if it's simply people moving into southern Britain, or if it's simply this churn, this homogenization across Europe that we're seeing reflected here. We're currently working on samples from a range of sites in Bronze Age and Iron Age France, and hopefully they'll start to give us some further information on that. But we have to think about the underlying social processes. Why are these people? What is creating this movement? Is it simply marriage networks? Is it small groups fissioning off and moving in search of better land? What are the reasons behind this? Why does the lactase persistence allele follow such a different trajectory in Britain from what it does in continental Europe? That's something that we really had no idea of at all. And now it's something that we have to try and think about, we have to try and explain. And crucially, I think the next stage of the project is going to be burrowing into this prehistoric kinship data. How do we actually interrogate kinship practice from genetic data? There are all kinds of problems and issues in trying to do that. The ethnographic record is exceptionally complex. Joe Brook published a paper quite recently about some of the range of approaches and attitudes to kinship that we see through the ethnographic record that is by no means restricted to biological relatedness. But at least now we can actually start to explore and investigate some of these patterns in a way that we really couldn't have imagined we could do 5, 10 years ago. Okay, I'm going to leave it there. Thank you very much.
Thank you very much, Ian. That gives us some real food for thought, I think. I'm particularly interested in this last bit you're tantalizing with us on over kinship in cemeteries, and clearly Wet Wang offers real fantastic potential for that. Does what you're getting at Wet Wang map onto the spatial organization of the cemetery?
It does to a partial extent. It's not completely obvious. The burials that we're seeing as having close family relationships or biological relationships to each other are broadly in the same part of the cemetery. They're quite close together, but we're not seeing really clear marking of that biological relatedness in the structuring of the tombs themselves. I mean, I think it very much is a teaser. We're going to get a lot more detail on this. One of the things we find, we did some work earlier last year on a Neolithic site led by Chris Fowler at Haslerton North, where we had lots of family relationships and went back and collected some more samples. And what you find with these things is that as soon as you have one or two more individuals, suddenly it starts locking together these family trees into much more robust form. So I think that once we have that higher level of data, we can then really start to interrogate some of the spatial pattern.
That really is fascinating. If you're doing such an intensive study of Wet Wang, it also has a very strong spatial structure, doesn't it?
Yeah,
and
absolutely.
That's really opening up.
Yeah. I mean, John Dent has done a huge amount of work on the sort of spatial, the growth of the cemetery from various points across it, and it'll be fascinating to see how that relates to these kind of generational movements.
Can I invite questions? We've clearly got some online. We'll take them.
This is a question that was asked when you were showing some of your slides beforehand. They say the samples seem skewed towards southern England rather than equally distributed across mainland Britain. Are there sufficient samples from outside southern England to reach definitive conclusions?
Yes and no. We have a big problem in northern England, essentially, simply with the survival of human remains, and we simply don't have enough samples north of Yorkshire, really, to say where the real change occurs. We have a lot of samples from Scotland, and so we can definitely say that what's happening in Scotland is different from what's happening in England and Wales. But whether there's any kind of sharp cutoff, I wouldn't imagine there is, but we can't see the gradation from one to the other because of the lack of samples in that intermediate period. We have got, not in that paper, but the samples that we're building up now, we are beginning to get some samples. We have one site in Cumbria, we have another site in Northumberland, but it's extremely patchy. And if anyone knows of any relevant Bronze or Iron Age human remains from that region, we would be very, very happy to have a look at them.
Someone asked, would you mind repeating the name of the Joe Brook paper you mentioned?
The name of the paper? I can't repeat it because I didn't say it.
Someone in chat's just replied to
Good.
ancient
It was
DNA, kinship, and relational identities.
That's the one. Yeah.
And one more, is more known about earlier Scottish outliers?
Earlier Scottish outliers. In the sense of the very early ones, in the Neolithic outliers, I presume that's probably what the question refers to because that's what was shown on the graphic. Basically, there are a cluster of individuals from northwest Scotland, a lot of them from cave sites, where we have much more hunter-gatherer ancestry than we can see anywhere else in Britain. And they're very, very interesting, and we're doing more work on the sites that they came from. So I'm hopeful we'll be able to say more about it in 6 months to a year. But it does look like that there is more of a kind of I guess more of a robust Mesolithic population in that region that's having much more of a genetic legacy, at least into the early Neolithic period, than we see anywhere else in Britain. The relevant paper is one that was published by large group of people, but Selina Brace was the main author in 2019, looking at the DNA underlying the arrival of the Neolithic in Britain. So all of the data is in that paper.
So a follow-up, in a sense, to the first question. If you've got more samples in Scotland and you're fairly confident, therefore, that that's a representative sample, if it is, but you're not so confident about the samples in England, how confident can you be about that chart that you put up about the EEF genetic history crossing over and being more pronounced in England later? And a sort of general question.
Yeah.
How confident are you? Are there checks that you can do that suggest that the samples you've got are typical of the time period?
Yeah, it's always a difficult question talking about sample sizes, because there was a lot of unease, I think, with the Beaker paper about sample sizes at that point. I think the thing to say is that an individual sample is not just telling you about that individual. That individual is actually telling you about potentially thousands of people within the community, their community of ancestors, essentially. So it really doesn't take an awful lot of samples to start to be really fair, to build a fairly robust interpretation. But in England, for the period that we're talking about, we have 400 or 500 samples from England. So it's simply that region in the north that we have a real problem with. Wales, we're not great on either. Again, it's the same problem of survival of human bone. But when we look at the individuals that we see in Scotland, and we look at the individuals that we see in England, south of Yorkshire and further south, there's a major difference between the two. And in fact, Nick Patterson, who led on the analysis in the paper, we've got samples we haven't published yet on early medieval Scotland, and those individuals genetically look like Beaker people. Essentially, there really is very little genetic turnover in pretty much all of Scotland, really, from the early Bronze Age into the early medieval period until the Norse arrived. But in England, it's very different. So we can be very sure of that. What we're doing now as we build up the numbers of samples is, the next stage is to start to look at population structure within Britain itself and look at the relationships between different regions. And that's already starting to show some quite interesting things, but we're not near publication of that.
Sorry. You mentioned language and linguistic analyses. I was wondering whether, well, what does that constitute? Is it place names or?
Oh, no. One of the things that there was a lot of debate in the team about the extent to which we should sort of start to speculate on things like language when we were publishing the genetic data. But the data certainly does impinge on the debate around the introduction of Celtic languages into Britain. Because traditionally, of course, there have been various theories, but one of the major ones has been that Celtic languages come in during the course of the Iron Age. Now, if language introduction is associated with spread of people, and that's the assumption that we have to make, if that's the case, then there is really nothing to suggest that Celtic languages can be coming in during the Iron Age. However, one of the other theories about the spread of Celtic languages that's been led mainly by Patrick Sims-Williams sees an earlier introduction, and I think he was talking about really a spread from France around the end of the Bronze Age. And our data fits very, very nicely with that interpretation of the spread of Celtic languages. So it may be that that's the period when we're seeing the spread of Celtic languages across quite great swathes of Europe related to that homogenization of population. I'm not 100% believer in the link, but it's certainly interesting, and it's something I think that we have to think about when we're thinking about language change at this period. There's an additional question about the introduction of Indo-European languages in the first place, and there's a school of thought now that would associate that with the spread of steppe ancestry at the beginning of the Bronze Age.
Thank you very much for that, Ian, for your paper on yourself and your team. Going back to Wet Wang Slack, and so having looking at all of those burials, it's going to be amazing what you can do when you include the isotopic work, but also the material within those graves, looking at sort of how varied some of those assemblages are, pulling together material from different locations. What I'm curious about, though, is those cemeteries are pretty special. You don't often get large clusters of dead bodies. So how many other assemblages have you got across the UK which do have lots of individuals where you're going to be able to do comparable work to see if the patterns of perhaps movement or relationships that you've got in Yorkshire is different elsewhere?
We have no comparable assemblages to East Yorkshire. I mean, in terms of the largest assemblages that we're looking at, which have been done since that paper was published, we have about, I think about 90 individuals from Danebury. We have Sudden Farm, which is a big assemblage of about 45, something like that, but nothing on the scale of Wet Wang Slack, because nothing exists that we've found on the scale of Wet Wang Slack. So the majority of our assemblages from the Iron Age are small, in the teams, of individuals. So no, we're not going to be in a position to replicate that work anywhere else. But I think we've probably going to have a work cut out with Wet Wang Slack for a bit in terms of trying to understand what's happening there.
Someone's asking, is it possible to get details of the paper you mentioned regarding robustness of Mesolithic in West Scotland?
Yeah, I can't remember the name of the paper off the top of my head, but it was by Selina Brace et al. 2019. It's called Ancient Genomes and something. So I'm sure a Google search will probably pull it up. I think it's published in Nature, Ecology, and Evolution.
Sorry, just in case it's the final answer, sorry, final question. You've called it the Commios project. Now, you've also used an illustration of an individual who was buried at Bursted, whose TVAS has claimed is actually Commios. Have you sampled him, and did he like milk?
We have sampled him, but we don't have the results yet. So whether he liked milk or not, I'm not sure. I don't think he's Commios, unfortunately. It'd be great if he was.
Best question is always saved till last. Ian, this is fantastic work. Thank you very much. Can we express our thanks again to Ian? Splendid paper. I would, as I've done several times in the last year, draw attention to the fact that the ERC is responsible for funding this and has been transformative in funding archaeological projects. I give notice that the next meeting on Thursday, the 17th November at 5 p.m. will be an unexpected complex of Roman buildings at Eastfield, Scarborough, by Paul Aware and Pete Wilson Fellows. Meeting stands adjourned.